Hi, my name is Dr. Mariella Philbin, and I'm a pediatric neuro-oncologist at Dana-Farber Cancer Institute and Boston Children's Hospital.
Thank you so much for supporting our high-risk, but hopefully high-reward work on pediatric high-grade glioma. As you probably know, these tumors are among the most lethal in our pediatric cancer population, and almost nobody survives beyond 1 or 2 years after diagnosis. Why that is, we really don't understand so far, but we do know that our current treatments do not work. So how can we find a completely new approach to target these tumors? One way is to look at not only the cancer cells or tumor as a whole, but also look at the interactions of the tumor cells with the normal cells that surround and feed them. And what we do to get a handle on this is single-cell RNA sequencing, which means we sequence every single cell within the tumor, and not only the tumor cells, but also the normal blood vessels, immune cells, and resident normal brain cells, to find out what's actually making the tumor comfortable in the brain, and how can we then interact or block these back and forth communication lines to find new treatments. One very exciting finding we noticed is when we looked at all the single cells from DIPG tumors or diffuse intrinsic pontine gliomas, was that all the tumor cells were not all the same either, meaning that some tumor cells were really bad in a way that they were stem cell-like, dividing a lot to produce more bad cells, but about a third of tumor cells within untreated patient tumors was able to differentiate or mature and become almost normal cells. They were still tumor cells because they had the same mutations and chromosomal abnormalities as all the other tumor cells, but their appearance and their behavior was much more like normal cells. They even looked like normal astrocytes or oligodendrocytes under the microscope. This finding was exciting in 2 ways. First of all, it means that even without any treatment, certain tumor cells figure out a way to become almost normal and be reminded of what they were supposed to do in the first place, which is to become a normal cell, stop to proliferate and divide, and start doing the job in the brain, so to say. But second of all, and this is very exciting to us, can we use this finding to therapeutic ends? Meaning that if some of these cells can already figure out a way to mature and stop dividing, can we push the remaining cells that way also? Even those cells that have forgotten what they were supposed to do. And thereby, in addition to just killing the tumor cells, differentiate the cells and use that as a new way for treating our children and young adults with these horrible tumors. The hope we have generated with these findings in our lab is very palpable, as we have now moved to the next step and indeed have already found a few genetic targets and also chemicals that target these genes that, when given to DIPG and GBM cells, can actually push cancer cells towards maturation. We believe that this could ring in a new phase of treatment for pediatric and young adult brain tumors, and we couldn't have done this without your support. In addition to this initial attempt of finding all the single cells within high-grade gliomas, we also recently started a project called Project HOPE. This is a fantastic multi-center national study on not only pediatric, but also adolescent and young adult patients with relapsed high-grade gliomas. In this project, we work with many researchers around the country to find out not only what drives the original tumor of these patients, but then also look at the same exact patient's tumor when it reoccurs after treatment and ask the questions, what has changed during treatment? And who are the resistant cells who are now there and gave rise to this recurrent tumor? And most importantly, again, are there any new programs or even new kinds of cells we now have to target that are different from the original tumor? This particular Project HOPE was only possible because of people like you who advocate for and support brain tumor research. Because big projects like this require not only collaborations across different institutions, in this case between Stanford, UCSF in San Francisco, CHOP in Philadelphia, and Dana-Farber Boston Children's Hospital, but it also takes coordinated funding, and was only possible because of coordinated funding between the National Brain Tumor Society and the National Cancer Institute. So thank you again for all your support. We could not do this without you. Let's fight brain cancer together and put an end to this disease.
Hi, everyone. Welcome to National Brain Tumor Society's webinar for Childhood Cancer Awareness Month. I'm David Aaron, CEO of National Brain Tumor Society, and this is a webinar called What is Project Hope? I'm honored today to have with me Dr. Mariella Philbin and Dr. Alfred Young to help us learn more about this research and why it is important to the brain tumor community. First, let's do introductions of our esteemed guests. Dr. Philbin is research co-director of pediatric neuro-oncology, Pediatric Neuro-Oncology Program, and assistant professor of pediatrics at the Harvard School of Medicine in Dana-Farber. Dr. Alfred Young is National Brain Tumor Society's distinguished scientific advisor, professor of neuro-oncology, and former chairman of neuro-oncology at University of Texas MD Anderson Cancer Center, and was a member of former Vice President Biden's Cancer Moonshots Blue Ribbon panel. Welcome to both of you, and thank you for being here with us. Together, you've been leaders in this project ever since its start, and by being here, you also represent a lot of other researchers involved in this project. In fact, researchers across our country and even internationally. I'd like to also say a special thank you to Dr. Ned Sharpless, Director of the National Cancer Institute, Dr. Lou Stout, and for the NCI's support and investment for this project. And their vision and their leadership is very much behind this as well. This project came about, as you know, and in the videos we've done, tell that story, is a great example of advocacy in action and how patients, caregivers, researchers, all working together to encourage the federal government, in particular Congress, to increase funding for the National Institutes of Health and the National Cancer Institute translates into more money, and then it allows the National Cancer Institute to make investment choices in the kinds of cancer that it's trying to advance. And of course, we know the NCI has put a flag on gliomas and aggressive brain tumors as a recalcitrant cancer, a stubborn cancer for which there's high unmet medical need, and we need to advance research much more quickly. While we've seen survival rates in other cancers improve, we need to do better in brain tumor. And this research represents a massive effort to do better and to do things quicker and to unravel the complexities of brain cancer. We all know, as you know, as treating physicians, we need breakthroughs now. Parents, patients are very frustrated because we need more treatment options and better ones. This is much more than about publishing papers. This is about ultimately producing products that work. This is about getting real-time potential impact for patients and families through better understanding the drivers of their disease at that molecular and indeed cellular level. And it starts with this kind of research. We can't get to better products without doing better biological research and better genomic research. And this project represents that, and I'm excited for you to share more about it. Hopefully, all of this will lead to safer, less toxic treatments and also improved quality of life if we do this correctly. Dr. Philbin, let's start with you. A few questions, and then we'll turn to Dr. Young and be able to have this discussion. Dr. Philbin, in reflecting upon Project Hope, what makes this research so important in terms of helping to develop the future of new therapies?
Thank you, David, for this question, and I'm very honored to be here with you and Dr. Young. And on behalf of our whole group of Project Hope and on behalf of all of our patients and families, we really also want to thank the NIH and Dr. Sharpless for this amazing support. We wouldn't have been able to do this without the support and the leadership provided. So thank you. And in terms of what this is supposed to do, this project is, we really hope to provide the fundamental roadmap how we could treat pediatric glioblastoma in the future. And why am I saying that? We've learned a few very important lessons in the last few years. One of them was that pediatric glioblastomas are different from adult glioblastomas. They have different mutations, and they are just different in terms of their growth patterns and their overall reoccurrence. And what we've also learned by doing single-cell genomics on those tumors to start with was that glioblastomas are shapeshifters. which means every single cell can become can come in many different shapes and forms over time. And that changes also with treatment. So we are faced with a tumor that is extremely resistant up front, but even the therapies we are giving are changing the tumor in a way that makes it even more resistant after some while. So what Project Hope really is trying to achieve is to look at the same patient's tumor at the beginning of the patient's journey and after receiving treatment, and compare those two tumor pieces side by side and looking at what are the shape-shifting events that have taken place. And then, provided those basics, then design new treatments for the future patients that come our way.
Thank you so much. And when you're thinking about that shape shifting, that's fascinating. That shows us that we're dealing with an enemy that can change its color. It can be transform over time. And as a layperson, take away from that how we have to grapple with that kind of shape-shifting complexity in order to get better treatments. What should parents and families who are facing pediatric brain tumors take away from knowing the kind of research challenge that you're taking on? What should they know about the importance of tissue collection and the importance of getting tissue that may come from ultimately their child to helping this kind of research?
Yes, we are extremely grateful to every single patient and every single patient family coming to us and all the centers who are part of Project Hope and consented and allowed us to donate some of their tumor tissue for our research. It's essential that they give this selfless gift in many ways. And by selfless, I mean it's not going to benefit their own patient journey, their own child, but it's beneficial for hopefully every single patient who comes after them. So I can't thank every single one enough, and I want to encourage people when they're asked at their particular center if they want to participate in research, ask all the questions you want to ask, get your answers, reach out to people in the field, but please participate if you feel comfortable doing so.
Thank you, and thanks to all the parents and pediatric patients for being so giving of your biospecimens that help research. Dr. Philbin, what do you see are some of the next important milestones in Project Hope as it proceeds forward?
Yes, thank you for that question too. We initially had to assemble our big team across the nation, as you've already said, and we've said in the video, it's Stanford, UCSF, CHOP, and us at Dana-Farber Boston Children's. And the first stage was really to collect all these samples. And what sounds like an easy process wasn't actually all that easy. It took, as I said, all these patients and families and coordinated research programs to get those samples. We have now finished the sequencing of all the samples. And the next step now is the analysis. And we are all very excited and looking forward to this next stage. As you probably can imagine, this is also not trivial because we're dealing with terabytes and terabytes of data as we sequence every single cell in all the different patients' tumors. But all of those data are now available, and we're just really excited to look and answer the questions that we had, which is how different are recurrent tumors from the original tumor in this shape-shifting aspect? And then can we distill down a few shape-shifting events that occur in every single patient across the nation and across the world, and then start targeting those? So the next stage, I think, is why we are really doing this.
Good. It sounds like the next 2 years of this will be very important. Just imagine you're 2 years from now. What do you think we should all expect will be your deliverable or readout 2 years from now if you're successful?
If we're successful, and I really have high hopes of being successful, we will present our data, and not just present, but share all of our raw data with any other scientist out there who wants to use them. And the presentation will include the analysis, and I'm really hoping to find the distilled product of a few, let's say right now it looks like 4 to 5 programs that are active in every single patient, no matter when in the treatment course, and no matter where that patient is from, and no matter if female or male or whatever else is the difference between our patients. And once we have those distilled 4 to 6 programs, then having the first hint towards targeted treatments. I'm really hoping to come up with that short list for you within the next 2 years.
Thank you. Thank you, Dr. Philbin. Dr. Young, let's turn to you. And this is an an extraordinary project that, from my understanding, has never been done before. What do you believe is the significance of that the National Cancer Institute, part of the National Institutes of Health, has invested in Project Hope?
So first, David, thank you for having me joining with the webinar. I mean, this is a really exciting project. If we go back, the Project Hope, is a parallel project for the adult tumor, which we call Project CARE. The project actually was initiated 2 years ago, in May 2018, when we have a roundtable discussion sponsored by NBTS and the National Cancer Institute. So we invited Dr. Sharpless to attend, to join in with the roundtable to discuss, the status of malignant brain tumor research, and especially ask a question: why are we not making better progress? Why we do not have a silver bullet, a successful treatment paradigm for adult and children with malignant brain tumor, particular high-grade glioma like GBM. The outcome of the discussion point to a couple, very specific reasons. One is, particularly, we do not know enough of the tumor, especially the tumor cells and the interaction with the tumor environment, including the immune environment. We also do not know enough about when we give the patient a drug, whether the drug get into the tumor and whether the drug actually hit the tumor, as well as the reaction to the drug from the tumor and the environment. And the only way to answer these questions is that we have to study the tumor from the patient, not just tumor from the animal in the laboratory. So, and that's where the emphasis come on, and how can we study the patient's tumor better? And I think we are very grateful to Dr. Sharpless. In response to this, to the roundtable, Dr. Sharpless give us a mandate and say, well, come up with a proposal. We will find money for you if you can come up with a proposal to answer this. For us to really get started on how to answer this question about understanding the tumor better and understand the pathway of the drug better. And so we put together the Project Hope, and we make a proposal and say, by doing single cell analysis, which will allow us to look at individual tumor cell, group of tumor cell, and the individual group of immune cell, and how many immune cells are there, how many type, whether they're active or whether they're suppressive, and how they interact with the tumor cell. So with this kind of analysis, we'll be able to understand the immune environment, the interaction of the immune cell with the tumor cell, and the interaction of the drug and why certain drug work, how it works. So, and because we're going to be studying tumor before treatment and tumor after treatment. So that will give us a longitudinal comparison of what happened to the tumor after the treatment, when it shrink, and then when it come back. And this is the big project. Dr. Sharpless provided the initial set of money to do a pilot project. So in the last 2 years, as Dr. Philbin described, we started with a pilot project, collecting a large number, a small number of tissue, so that we can demonstrate that we can do it, and we can demonstrate all the technique. We can demonstrate that we can collect the sample. We can demonstrate that, and also start getting ready with the different platform of data analysis. These are all being done in the last 2 years. So now we're ready to expand the project because with a small number of tumor, we really don't have enough data to really draw specific and also definitive conclusion can help us to develop drug and develop treatment strategy. So we need a larger number of tumors. That means we have to expand the project. And this is again where NCI has to come in, because NCI will be able to provide the kind of leadership and provide the kind of resources that we need. And it's really important that you can get NCI behind us to support this.
Thank you, Dr. Young. Sounds like there's four things really coming together. One is the scientific ideas that, and Dr. Philbin, you laid those out of what are you trying to do with the science. Then we have this patient advocacy role and government coming together to provide the support and the investment. And we also know from the video and from this project, there's been unprecedented collaboration in Project Hope amongst leading pediatric neuro-oncology researchers across institutions. And then I wanted to ask you both about really another aspect of the parents' role in this is, what do you think the take-home message should be to patients, in this case parents, about the importance of what we call molecular profiling of brain tumors, of in every child or every patient, making sure that there is developing in pathology an understanding of that tumor. What's the relevance of that to contributing to this type of research, which is trying to unravel the complexities of brain tumors? And start with you, Dr. Philbin.
Yeah, thank you very much. As a clinician scientist, I have to say there's always two souls inside of me, and I have to speak for both sides. So the clinician side of me says that there's only a few genetic alterations that we know of right now that if you find them are targetable, meaning there is a drug available for those patients right now in September 2020. Whereas most of the tests we are doing are interesting in many ways, but we're just not quite there yet. So in that sense, they are research. And I'm always trying to balance that when I talk to patients and their families, what's really needed in order to make the diagnosis, the correct diagnosis for the tumor your child has, and what can we do in addition to make sure the field advances. I think both of those sides are equally important, but should be discussed with the treating physician or treating pathologist in their center.
Thanks. Thank you, Dr. Feldman. Dr. Young, what I've always learned from you over the years is the more you know your tumor at the patient level, at the doctor level, the better it is for patients and the better it is for research. Is that still the way it is? And does that help this kind of project?
Well, I mean, if you look at it, the reason why we need to know the tumor better is to really find a way to attack this tumor. And if we can find a way to attack the tumor, it's good for the patient, because it's the only way that we can prolong the survival for our patient and give our patient a better quality of life, is that we can find better treatment, and also very specific treatment that we can only treat the tumor and not treating the normal cells surrounding the tumor. And in our case, we don't want to damage the normal brain.
That's right. So
if we know the tumor and the environment, we can avoid damaging the normal brain. And that will give our patient, especially children, not only longer survival, but better quality of survival, better quality of life. So we need to go hand in hand. Look at the tumor, will be good for us to develop treatment, but also good for our patient, because we can improve not only survival, but quality of survival.
Thank you.
And I want to just add to that, sorry to interrupt, David. I wanted just to add to this that there's an incredible amount of hope currently in all the laboratories, not just ours here, but across the nation and the world, because there's so much new knowledge on how is glioblastoma or high-grade glioma growth really driven. And it's really about the immune system. It's about the normal brain cells, the nerve cells stimulating brain tumor growth. And it's also about the new targets that we've found. But a lot of that has not quite made the way into the clinic. But I'm thrilled, though, that I'm currently in the position that, in the next few years, I know those treatments will become available. Yeah. And it's an extremely exciting time for the whole field to finally move forward in the right direction.
Let's round this out while we have just a few minutes left with that treatments become available phrase, because there's another stakeholder group to make that happen, and that's industry, that's biotechs and biopharma. And what, Dr. Philbin, in your view, what should they hope for and believe is coming to them in terms of value from Project Hope? Why should they care about this project?
Yeah, I want to ask all industry partners in the future that if we approach them, that they would at least consider our plea to have a big heart for children with pediatric tumors, because we know that those numbers are much, much lower than for the adults. But the life years lost because of children losing their battle against cancer are immense. So really hope that our industry partners know that and are in it together with us and are willing and happy to partner with us once we present them our findings from this study.
Thank you. Well, it's almost the end of Childhood Cancer Awareness Month 2020. And we know that what we're all trying to do here is stop pediatric brain tumors from being the leading cause of cancer death in kids. We don't want to be number one anymore. We want to change the scorecard. And so thanks to your research, Dr. Philbin, your leadership and career of research, Dr. Young, I think we have a much better chance than ever at changing that scorecard. And so wanted to invite each of you to say one more sentence about your wish for Childhood Cancer Awareness Month 2020.
Yes, my one wish would be more a thank you to all the people who are already involved, and those include advocates and patients and the families, and my research colleagues and clinicians and industry partners and the government, because COVID 2020 or 2019 did not stop childhood cancer at all. And knowing that and seeing those patients every single day just reminds me that we have to fight even harder to end it, just like you said. So my wish and my plea is to keep on doing your great work, everybody, and together we will fight this disease.
Thank you. Dr. Young.
Well, we have a team of talented and motivated scientists, clinicians, working together, even day after day, without stopping by the COVID situation. And I hope, and I'm sure, we will make progress, and we will discover better treatment, and that's my biggest wish, that we can discover the specific effective treatment that can target the tumor and improve the survival and improve the quality of life of our patient.
Well, on behalf of National Brain Tumor Society, thank you for your innovation, your leadership, your collaboration, and most of all, your caring for patients and their families, in this case, children who are our most vulnerable patients. And I wish you all the speed in the world to make Project Hope and Project Care a great success over the next 2 years. We're counting on you, and we'll do everything we can to support this work. Thank you so much.
Well, thank you for your leadership, David, from the advocacy side.
And thank you very much from my side as well, and everybody who has participated in this wonderful project. Thank you.
Thank you.